11 Chapter 16 Excretory product and their elimination. Animal accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, calcium, phosphate, sulfate, etc. either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with the special emphasis on common nitrogenous waste. Ammonia, urea, uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia in ammonotalism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia is, as it is readily soluble, is generally excreted by diffusion across body surfaces or through gill surface in fish. As ammonia ions kidney do not play any significant role in its removal, excretory adaptation necessitate the production of lesser toxic nitrogenous waste like urea, uric acid for conservation of water. Mammals, many terrestrial amphibians, and marine fishes mainly excrete urea and are called urotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom present a variety of excretory structures. In most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidney. Some of these structures are mentioned here. Protonephridia or flame cell are the excretory structure in platyhelminthes flatworms. Example: Planaria rotifers. Some annelids and their cephalochordates. Amphioxus. Protonephridia are primary concerned with ionic and fluid volume regulation. Nephridia are the tubular excretory structure of earthworm and other annelid. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structure of most of the insects, including cockroach. Malpighian tubule help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function. Crustaceans like prawns. 16.1 Human excretory system. In humans, the excretory system consists of a pair of kidney, one pair of ureters, a urinary bladder, and a urethra. Figure 16.1. Kidney are reddish brown bean-shaped structure situated between the level of last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Toward the center of the inner concave surface of the kidney is a notch called hilum, through which ureter, blood vessel, and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis, with projection called calcae. The outer layer of kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calcaeous calyx. The cortex extend in between the medullary pyramid as renal columns called column of Batanae. Figure 16.2. Each kidney has nearly 1 million complex tubular structure called nephrons. Figure 16.3, which are functional unit. Each nephron has two types, the glomerulus and the renal tubule. Glomerulus are tuft capillaries formed by the errant arteriole, a fine branch of renal artery. Blood from the glomerular is carried away by an efferent arteriole. The renal tubule begin with a double-walled cup-like structure called Bowman's capsule, which enclose the glomerulus. Glomerulus, along with Bowman's capsule, is called the Malpighian body or renal capsule. figure 16.4, the tubule continue further to form a highly coiled network. Proximal convoluted tubule PCT, a hairpin shaped Henle loop, is the next part of the tubule which has a descending and an ascending limb. The ascending limb continue as another highly coiled tubular region called distal convoluted tubule DCT. The DCT of many nephrons open into a straight tube called collecting duct, many of which converge and open into a renal pelvis through medullary pyramid in the calcaire. The Malpighian capsule PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medullary. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephron. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephron. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called pertubular capillaries. A minute vessel of this network runs parallel to the Henle loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephron. 16.2 Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that take place in different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of the blood is filtered by the kidney per minute, which constitutes roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers: the endothelium of the glomerular blood vessel, the epithelium of the Bowman's capsule, and a basement membrane between these two layers. The epithelial cell of Bowman's capsule, called podocyst, and arranged in an intricate manner so as to leave some minute space called filtration slits or slit pores. Blood is filtered so finely through these membranes that almost all the constituent of the plasma except the protein pass on to the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The amount of the filtrate formed by the kidney per minute is called glomerular filtration rate, GFR. GFR is a healthy individual is approximately 125 ml per minute, 180 liter per day. The kidney have built-in mechanism is carried out by the juxtaglomerular apparatus, JGA. JGA is a special sensitive region formed by the cellular modification in the distal convoluted tubule and the afferent arteriole. At the location of their contact, a fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow, thereby GFR back to normal. A comparison of the volume of the filtrate formed per day, 180 liter per day, with that of the urine release, 1.5 liters, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubule. This process called reabsorption. The tubular epithelial cell in different segment of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acid, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by the passive transport. Reabsorption of water also occur passively in the initial segment of the nephron, figure 16.5. During urine formation, the tubular cells secrete substances like hydrogen and potassium and ammonia into the filtrate. Tubular secretion is also an important step in the urine formation, as it helps in the maintenance of ionic and acid-base balance of body fluid. 16.3 Function of the tubules. Proximal convoluted tubule PCT. PCT is lined by a simple cuboidal brush border epithelium, which increase the surface area for reabsorption nearly all the essential nutrient, and 70 to 80% of the electrolysis and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluid by selective secretion of hydrogen ions and ammonia into the filtrate and by absorption of HCO3 from it. Henle loop reabsorption minimum in its ascending limb. However, the region play a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of loop is Henle is permeable to water but almost impermeable to electrolytes. This concentrates the filtration as it moves down. The ascending limb is impermeable to water but allows transport to electrolytes actively to passively. Therefore, as the concentrated filtrated pass upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Distal convoluted tubule (DCT) condition reabsorption of sodium and water take place in the segment. DCT is also capable of reabsorption of HCO3 and selectively secretion of hydrogen and potassium ions and NH3 to maintain the pH of and sodium potassium balance in blood. Figure 16.5 reabsorption and secretion of major substance at different parts of the nephron. Arrow indicate direction of the movements of material. Collecting duct. The long duct extends from the cortex of the kidney to the inner part of the medulla. Large amount of water could be reabsorbed from the region to produce a concentrated urine. This segment allows passage of small amount of urea into the medullary interstitium to keep up the osmolarity. It plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of hydrogen and potassium ions. Figure 16.5. 16.5 mechanism of concentration of the filtration. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta plays a significant role in this. The flow of filtrate in the two limbs of Henle loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also in a counter current pattern. The proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium from 300 mO small in the cortex to about 1200 mO small in the inner medulla. The gradient is mainly caused by sodium chloride and urea. Sodium chloride is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amount of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of substance facilitated by the special arrangement of Henle's loop and vasa recta is called the counter current mechanism. Figure 16.6. The mechanism helps to maintain a concentration gradient. In the medullary interstitium, presence of such interstitial gradient help in an easy passage of water from the collecting tubule. By concentrating the filtration urine, human kidney can produce urine nearly 4 times concentrated than the initial filtrate form. 16.5 regulation of kidney function. The functioning of the kidney is efficiently monitored and regulated by the hormonal feedback mechanism involving the hypothalamus, JGA, and to a certain extent the heart. Osmoreceptor in the body are activated by exchange in body fluid volume and ionic concentration. And excess loss of fluid from the body can activate these receptors, which stimulates the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitate water reabsorption from later parts of the tubule, thereby preventing diuresis. An increase in body fluid volume can switch off the osmoreceptor and suppress as ADH released to complete the feedback. ADH can also affect the kidney function by its constrictory effect on blood vessel. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. GA play a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR, can activate the JG cells to release renin, which convert angiotensinogen in blood to angiotensin 1 and further to angiotensin 2. Angiotensin 2 being a powerful vasoconstrictor increase the glomerular blood pressure and thereby GFR. Angiotensin 2 also activate the adrenal cortex to release aldosterone. Aldosterone cause reabsorption of sodium and water from the distal part of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as renin angiotensin mechanism. An increase in blood flow to the atria of the heart can cause the release of atrial natriuretic factor ANF. ANF can cause dilation of blood vessel and thereby decrease the blood pressure. ANF mechanism therefore act as a check on the renin angiotensin mechanism. 16.6 micturition. Urine formed by the nephron is ultimately carried to the urinary bladder where it is stored till the voluntary signal is given by the central nervous system CNS. The signal is initiated by the stretching of the urinary bladder. It gets filled with urine. In response, the stretch receptor on the walls of the bladder sends signal to CNF. The CNF pass on motor message, initiate the concentration of smooth muscle of the bladder and stimulate simultaneously relaxation of the urethral sphincter, causing the release of urine. The process of release of urine is called micronutrition, and the neural mechanism causing it called the micturition reflex. An adult human excrete an average 1 to 1.5 liter of the urine per day. Urine formed is a light yellow colored watery fluid which is slightly acidic, pH 6.0, and has a characteristic odor, and on an average 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristic of urine. Analysis of urine help in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, glycosuria, and ketone bodies, ketonuria in urine are indicative of diabetes mellitus. 16.7 Role of other organ excretion. Other than the kidney, lung, and liver and skin also help in the elimination of excretory waste. Our lung remove large amount of carbon dioxide, approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secrete bile containing substance like bilirubin, biliverdin, cholesterol, degraded steroid hormone, vitamins, and drugs. Most of these substances ultimately pass out along with digestive waste. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat gland in the watery fluid containing NaCl, small amount of urea, lactic acid, etc. Through the primary function of sweat is to facilitate a cooling effect on the body surface. It also helps in the removal of some of the waste mentioned above. Sebaceous glands eliminate certain substances like sterols, hydrocarbons, and waxes through sebum. This secretion provides us protectively oily covering for the skin. Do you know that a small amount of nitrogenous waste could be eliminated through saliva too? 16.2 16.8 Disorders of the excretory system. Malfunctioning of kidney can lead to accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by the process called hemodialysis. During the process of hemodialysis, the process drained from the convenient artery is pumped into a dialysing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysis unit after adding a anticoagulant like heparin. The unit containing a coiled cellophane tube surrounded by a fluid dialysing fluid having the same composition as that of the plasma except the nitrogenous waste. The porous cellophane membrane of the tube allow the passage of molecules based on concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The cleared blood is pumped back to the back body through a vein after adding antiheparin to it. This method is a boon for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of acute renal failure, kidney failure. A functioning kidney is used in the transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such a complicated technique. Renal calculi, stone or insoluble mass of crystallized salts, oxalates, etc. formed within the kidney. Glomerular nephritis, inflammation of glomerular kidney. Summary: many nitrogen containing substances, ions, carbon dioxide, water, etc., that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretory vary among animals, mainly depending on the habitat. Availability of water, ammonia, urea, uric acid are the major nitrogenous waste excreted. Protonephridia, nephridia, malpighian tubule, green gland, and the kidney are the common excretory organs in animals. They not only eliminate nitrogenous waste but also help in the maintenance of ionic and acid base balance of body fluid. In humans, the excretory system consists of one pair of kidney, a pair of ureter, and a urinary bladder and a urethra. Each kidney has over a million tubular structure called nephron. Nephron is a functional unit of kidney and has two portions: glomerular and renal tubule. Glomeruli is tuft of capillaries formed from afferent arterioles, fine branches of renal artery. The renal tubule start with a double-walled Bowman's capsule, and it's further differentiated into a proximal convoluted tubule PCT, hence loop HCl, and distal convoluted tubule DCT. The DCT of many nephrons join to a common collecting duct, many of which ultimately open into a renal pelvis through a medullary pyramid. The Bowman's capsule encloses the glomerulus to form Malpighian or renal capsule. Urine formation involves three main steps: filtration, reabsorption, and secretion. Filtration is a non-selective process performed by the glomerulus using the glomerular capillary blood pressure. About 1200 ml of blood is filtered by the glomerulus per minute to form 125 ml of filtrate. The Bowman's capsule per minute GFR. JGA, a specialized portion of the nephrons, play a significant role in the regulation of GFR. Nearly 60% of the reabsorption of the filtrate take place through different parts of the nephron. PCT is the major site of reabsorption of selective secretion. HL primarily help to maintain osmolar gradient 300 mmol minus 1200 mmol within the kidney interstitium. DCT and collecting duct allow extensive reabsorption of water and certain electrolytes, which help in osmoregulation. Hydrogen, potassium, and NH3 could be secreted into the filtrate by the tubules to maintain the ionic balance and pH of the body fluid. A counter-current mechanism operates between the two limbs of the loop of Henle and those of the vasa recta capillaries. Parallel to Henle loop, the filtrate gets concentrated as it moves down the descending limb but is diluted by the ascending limb. Electrolytes and urea are retained in the interstitium by the arrangement. DCT and collecting duct concentrate the filtrate about 4 times from 300 mmol to 1200. An excellent mechanism of conservation of water. Urine stored in the urinary bladder till a voluntary signal from CNS carries out. It's released through urethra. Micturition, skin, lung, and liver also assist in secretion.